Photosynthesis is one of the most important biological processes on the planet. Most complex life forms rely on photosynthesis for energy-giving carbohydrates and oxygen. We mostly associate photosynthesis with plants, but in reality, other eukaryotic organisms like algae and even prokaryotes can also perform photosynthesis. In fact, most of the world's photosynthetic organisms are prokaryotic and, together with the algae and plankton from the oceans, contribute to the majority of the planet's oxygen reserves. In this video, we will be giving a summary of photosynthesis in the context of bacterial metabolism. Specifically, we will be talking about phototrophy, an overview of photosynthesis, photosynthesis in bacteria, light and dark reactions, some recent advancements in the scientific field related to photosynthesis, and we'll be answering some interesting questions as well. By watching this video, you will be able to learn about photosynthetic pigments and where they are located in prokaryotes and eukaryotes. Compare and contrast the light-dependent from the light-independent reactions. Describe the Calvin-Benson cycle. Differentiate between cyclic and non-cyclic photophosphorylation, and give some examples of photosynthetic bacteria. So sit back, relax, and put on your learning hats, because today we will be talking about photosynthesis. Before we get into a detailed view of what happens in photosynthesis, it is important to first establish a general understanding of it. So, in its essence, photosynthesis is the process wherein plants, algae, photosynthetic bacteria, plankton, and other organisms harness light energy from the sun and convert it into chemical energy. The general equation for photosynthesis is 6 carbon dioxide molecules plus 6 water molecules forms energy in the form of glucose and 6 oxygen molecules. So, through photosynthesis, phototrophic organisms can convert the inorganic molecules of carbon dioxide and water into glucose and oxygen, both of which are essential for a lot of organisms to survive. Photosynthesis can be divided into 2 stages: the light-dependent reactions and the light-independent reactions. Both of these will be discussed in more detail later on, so this part will just give you an overview. The light-dependent reactions, or the light reactions, are driven by, you guessed it, light. This means it cannot occur without the presence of photons from sunlight. In this stage, photons are used to split water, eventually generating oxygen, ATP, and NADPH or NADH. These energy-carrying molecules are then used in the light-independent reactions, or the dark reactions, to drive the energetically unfavorable process of assembling sugar molecules from carbon dioxide. The dark reactions are called dark, although they still occur in light because they do not depend on sunlight for them to occur. The dark reactions do not immediately produce glucose. Instead, it produces PGAL, or phosphoglyceraldehyde, also known as G3P, or glyceraldehyde triphosphate, which is a 3-carbon molecule that is used to make glucose, which is a 6-carbon molecule. For photosynthesis to occur, certain structures must be present in prokaryotes and eukaryotes to enable the transformation of light into chemical energy. In eukaryotic cells, photosynthesis happens inside a chloroplast, a unique organelle enclosed by a double membrane. The endosymbiotic theory suggests that these organelles may have arisen when a bacterium engulfed a photosynthetic bacterium, but instead of digesting it, they learned to live together in endosymbiosis. Inside the chloroplast are disc-shaped photosynthetic structures called thylakoids. These thylakoids usually form stacks called grana, while the space surrounding the granum in the chloroplast is called the stroma. Prokaryotes, on the other hand, do not have membrane-bound photosynthetic organelles. Instead, they have infoldings in their plasma membranes, which serve as the photosynthetically active regions. In the case of cyanobacteria, these infolded regions are also known as thylakoids, and within these thylakoid membranes and other photosynthetic bacteria membranes are photosynthetic pigment molecules organized into photosystems where light is converted into chemical energy.
Before we dive in further to the process of photosynthesis, let us first understand the process of phototrophy, which literally means light eating. It is a process by which energy from the sun is captured and converted into chemical energy in the form of ATP, whereas photosynthesis is more precisely the process of converting inorganic materials like carbon dioxide by using the energy product of phototrophy into usable organic compounds. Two types of organisms undergo phototrophy, the photoautotrophs and the photoheterotrophs. Photoautotrophs are also the photosynthetic organisms that use light and carbon dioxide for their sustenance, while photoheterotrophs are organisms that also use light but obtain their carbon source from organic compounds produced by other organisms or from their environment. But to convert the solar energy into usable chemical energy, molecules sensitive to light called pigments come into play. The popularly known pigment which is used by plants, algae, and cyanobacteria is the chlorophyll, a molecule that absorbs blue and red light at around 430 nanometers and 675 nanometers respectively, which transmits a green color. In purple and green bacteria, they have bacterial chlorophylls that absorb higher wavelengths than the chlorophylls at around 870 nanometers. Aside from this, there are also accessory pigments that expand the range of light wavelengths that can be used, such as carotenoids, phycobiliproteins, which is divided into phycoerythrin and phycocyanine. In this way, the available light is maximized and allows different phototrophs to occupy the same environment without competing with one another. The accessory pigments also act as antioxidants that serve to protect the organism. These photosynthetic pigments are organized into photosystems, which are reaction centers composed of light harvesting complexes called photosystem antennae. There are two types, photosystem 1 and photosystem 2, which are excited by light energy initiating light-dependent reactions, which will be further discussed later on. Both PS1 and PS2 can be found in plants and cyanobacteria, whereas only one of the two photosystems is used by the anoxygenic photosynthetic bacteria. Moving on, let's tackle bacterial photosynthesis, and we'll only be focusing solely on photoautotrophs. A brief history: photoautotrophs have been existing long before the Earth's atmosphere could sustain human life, but only in recent years have scientists begun to research these microorganisms regarding their photosynthetic mechanisms. Here is an animation on the mechanism of bacterial photosynthesis on a purple bacterium, where Pseudomonas viridis. R. viridis consists of a reaction center embedded in its membrane that pumps protons across the membrane. Within its reaction center protein lie the pigments and several electron carriers. The photosynthetic pigments in this bacterium is called bacterial chlorophyll. This chlorophyll absorbs a photon from a light source that excites an electron. This excited electron is then transferred to one of the accessory proteins, phaeophytin, making the chlorophyll positively charged. It is then passed to another accessory pigment called menaquinone, exciting it again, then to another called ubiquinone. Ubiquinone carries two protons within two added electrons across the membrane to deposit those two protons on the other side of the membrane. Simultaneously, the two electrons are carried by mobile cytochrome back to the chlorophyll to restore to a ground state ready for another cycle. Hence, this is called a system of cyclic phosphorylation, forming ATP and reducing NAD. Going back to photosynthetic bacteria, It has two classifications: oxygenic and anoxygenic photosynthetic bacteria. Oxygenic photosynthetic bacteria operates photosynthesis similar to plants. They have light harvesting pigments, absorb carbon dioxide, and release oxygen. Cyanobacteria or cyanophyta are the only known oxygenic photosynthetic bacteria. They are usually found in the water but can also survive on land, in rocks, animal shells or fur, and in corals. This is the general photosynthetic reaction of cyanobacteria. Very similar to plants, right? Now, anoxygenic photosynthetic bacteria on one end also consume carbon dioxide but do not release oxygen. They release sulfur instead, in the case of sulfuric bacteria. They are found below the surface of lakes and ponds where oxygen content is low. Sulfur is reduced and other necessary compounds are available. These are the different types of anoxygenic photosynthetic bacteria. First, we have the green sulfur bacteria such as the chlorobion species and the Chlorosudomonas species, which has the bacterioviridine pigment, and this is its reaction. Next, we have the purple sulfur bacteria such as the Thiospirillum species with the pigment bacteriochlorophyll and carotenoids, and this is its reaction. Next is the purple non-sulfur bacteria such as the Rhodospirillum species and the Rhodosudomonas species. The pigment they have is the bacteriochlorophyll, and this is its reaction.
There are two stages in photosynthesis. The first stage involves light-dependent reactions or phosphorylation. So, what happens during light-dependent reaction? First, it is important to know that phosphorylation yields oxygen, adenosine triphosphate or ATP, and nicotinamide adenine dinucleotide phosphate hydrogen or NADPH. Phosphorylation can proceed in two ways: non-cyclic and cyclic. Non-cyclic phosphorylation releases electrons that would then be incorporated into the NADPH molecule and do not return back into the system. In contrast, in cyclic phosphorylation, the electrons released from the photosynthetic pigments will eventually return to the system, and no release of electrons happen. We made these simple animations to show an overview of what happens during the reaction. The first one shows non-cyclic phosphorylation. The process starts with two electrons derived from the splitting of water, and that supplies the electrons to photosystem 2. Then, light energy in the form of photons is absorbed by the chlorophyll molecules. This then excites the electrons in photosystem 2. The excited electrons are then carried by the mobile carrier phostoquinone QB to cytochrome B6F. Having lost two electrons, photosystem 2 acquires two new electrons through the splitting of water molecules. From cytochrome B6F, the electrons are then carried to plastocyanin, another mobile carrier, into photosystem 1. As light energy hits photosystem 1, the electrons get propelled towards ferredoxin NADP reductase, or FNR. These two electrons, along with the hydrogen ion, are added to NADP, creating NADPH. This process is also known as non-cyclic phosphorylation. The process allows for the creation of an ATP gradient, which is then used by the ATP synthase to create ATP from ADP. Cyclic phosphorylation undergoes almost the same process, but the electrons return back to photosystem 2 to be used again in the cycle. This cycle allows for the formation of ATP.
Now, We move on to the second stage of photosynthesis, known as the light-independent reactions, or the Calvin cycle. These reactions take place at the stroma of the chloroplast, wherein fixed carbon atoms are used to build three carbon sugars. Although the reactions do not directly depend on light, it is dependent on the ATP and NADH produced in the light reactions. So what happens during the Calvin cycle? There are three main stages involved in the Calvin cycle: carbon fixation, reduction, and regeneration of the starting molecule. In the first stage, which is carbon fixation, we need two additional components to initiate the reaction: the enzyme ribulose bisphosphate carboxylase, or RIBISCO, and three molecules of ribulose 1,5-bisphosphate, or RuBP. For each carbon dioxide molecule that reacts with RuBP, two molecules of 3-phosphoglyceric acid, or 3-PGA, are formed. So this reaction produces 6 molecules of 3-PGA. In the second stage, which is reduction, the 6 molecules of 3-PGA are converted into 6 molecules of a 3-carbon sugar, glyceraldehyde 3-phosphate, or G3P, by ATP and NADH. Let us recall that in reduction, an electron gains an atom or a molecule. This stage is a reduction process since NADPH donates electrons to the 3-carbon intermediate to make G3P. In the last stage, which is regeneration, only one of the G3P molecules leave the Calvin cycle to make glucose, while the remaining 5 are recycled to regenerate the RuBP acceptor. This stage requires 3 more molecules of ATP for it to take place. The G3P molecules have 3 carbon atoms, so we will need 3 carbon dioxide molecules to enter the cycle to provide 3 new atoms of fixed carbon. Thus, it takes 3 turns of the Calvin cycle to release 1 G3P.
Plants have this extraordinary ability of converting sunlight into energy. Yes, they can make food from sunlight, but they're not really the most efficient at doing this, especially in the storage aspect of things. Imagine being able to convert a large percentage of solar energy into usable energy, store it for a period of time, and use it whenever necessary. Cool, right? Professor Nate Lewis and Dan Norcera thought so too. That is why they sought to invent the artificial leaf. Nate Lewis's preferred equipment, an integrated solar fuel generator. His equipment is able to do what plants can do, only better, by using a better way of harvesting light, where its 2 photoelectrodes can absorb sunlight using 2 different colors of the spectrum, allowing for a more efficient way of obtaining energy. The process then proceeds just like what plants would do: split water, use catalysts to speed up the half reactions, then obtain energy to be used to do a variety of things, from fueling vehicles to generating electricity for the grid, and to serve as feedstock for the generation of other complex fuels. Dan Norcera, on the other hand, is a scientist from Harvard University that is also aspiring to commercialize an artificial leaf. However, in contrast to Lewis's main idea, which is to produce hydrogen, Norcera aims to bypass the intermediate production of hydrogen and develop a device that uses sunlight to directly produce carbon-based liquid fuels, which can immediately replace petroleum products. Yes, the idea sounds a bit futuristic, but if it could be commercialized, done cost-effectively, and at a scale, it would be a very efficient method of storing sunlight in the most versatile fuels today.
Did you know that X-rays and UV lights are harmful to the skin? They use ionizing radiation that causes radiation-induced deterministic skin effects that range from mild effects such as transient erythema or temporary epilation to severe effects such as desquamation and necrosis. X-rays, solar radiation, UV-emitting tanning devices, and UV radiation are considered carcinogens by the World Health Organization. High exposure to UV rays can cause sunburn, premature aging of the skin, and signs of sun damage such as wrinkles, leathery skin, liver spots, actinic keratosis, and solar elastosis. Did you also know that when green algae are exposed only to UV light, they would not be able to perform photosynthesis? Only the visible light range from red to blue are photosynthetic radiation. Since UV light has too much energy, it is not viable for photosynthesis. UV light causes the cell wall of algae to break, causing the algae to clump together and die. Photosynthesis is an essential process for our ecosystems and the biosphere as a whole. It keeps our ecosystem running through the exchange of the products, the transformation of energy, and the cycling of nutrients. It is also an essential process that can help mitigate the effects of climate change. Thus, it is important to keep these photosynthetic organisms around as they also help maintain biodiversity. Just a quick review. In this lesson, we learned about phototrophy and how pigments are involved in the transformation of light energy to chemical energy. We also differentiated light-dependent and light-independent reactions. In the light-dependent stage, we learned about the difference of cyclic and non-cyclic phosphorylation and how they produce ATP. And then, how it proceeds to the second stage of photosynthesis, the light-independent Calvin cycle, which undergoes carbon fixation, reduction, and regeneration of ρBP, in which after 3 cycles produce 1G3P. And since we were talking about light and electromagnetic radiation, we also learned the harmful effects of X-ray and UV light on the human skin, and that green algae can perform photosynthesis with UV light alone. We hope you had fun, and we'll see you in the next one. Until then, stay curious.